Thank you for being here. So I want to talk about our genome mapping technology to look at structural variation in the genome. And so mapping, what does that really mean? Well, I need to start by saying that Bionano is not a sequencing technology. We don't read the A, C, Gs, and Ts of the genome. And there's good reasons for not doing that, and I will show you what these reasons are. If you're here for sequencing, I recommend that you check out the workshop next door. But so what is the problem? Why does sequencing not give us all the answers about the genome when it comes to structural variation? Well, the genome is just really highly repetitive. Just about a third of the genome, that gray section here, consists of unique sequences, and 2/3 of the genome consists of repeats. And our most major structural variations, insertions, deletions, duplications, translocations, and inversions are often mediated by repeats that recombine with non-allelic homologous recombination. And so if you use a sequencing technology that gets bogged down in these repeats, then you're often blind to the structural variants that are flanked by these repeats. So Bionano looks at the genome in a very different way. We image megabase-sized molecules. The shortest that we typically use are 150 kb, and we go all the way up to 3 megabase pairs. And this is what our raw data looks like. Very long molecules. The average molecule length on our system is about 300 kb. And we label a single 6 base pair sequence motif all throughout the genome. A single sequence motif that is uniform enough throughout the genome that we really don't have any blind spots, and with a label density that's high enough that we have a high resolution to detect structural variants. So we actually observe structural variants directly in the patterns on these molecules. And then we just stain the backbone, and that gives you that blue signal. Now I'll talk real quick about how it technically works. The first step to image megabase-sized DNA is, of course, having megabase-sized DNA. And that's not obvious, because every major standard method to isolate DNA fragments it at about 30 or 50 kb or so. And that's true whether you use spin columns, whether you use magnetic beads that grind the DNA apart, or simply precipitate it, and these forces also shear the DNA. So Bionano gets around that by using agarose gel plugs. It's a fairly old-fashioned system, but it works really well. You embed the cells or nuclei in a little cube of agarose, purify the DNA with enzymes and detergents, and then you digest the agarose away once the DNA is clean. And chromosome R-length molecules come out. This works really well, and we have protocols mostly used now for tissue, animal, of course, human tissue as well. And for cancer biopsies, we can go as low as 10 milligrams. Now, it's important here to know that FFPE samples won't work. The DNA is just too fragmented. Now, just recently, we introduced a new DNA isolation protocol that's much faster. It's a solution-based protocol. And here we simply lyse the cells in solution and grab onto them with a disc made out of a material very similar to spin columns. And then you simply take the DNA out, put it in a clean tube and leave all the debris behind. It's very fast, takes only about 4 hours or so versus 2 days with agarose plugs. It's automatable, and we're working on that. And we can use this protocol right now for fresher frozen blood. We need about 600 microliters, and about 1.5 million cultured cells work as well. We have very clean DNA, and it works just beautifully. So now we have megabase-sized DNA. We label a single sequence motif all throughout the genome, and we used to do that with nicking endonucleases, but about 1.5 years ago, we introduced our direct labeling chemistry that in a single enzymatic reaction attaches a fluorophore directly to that 6 base pair recognition sequence. There's no more nicking or breaking of the DNA, no more blind spots, and our data has gotten a lot better through that process. We load that labeled DNA, and now it looks like this, coiled up, very long molecules with a fluorescent label. And we load that into our chips. And our core technology really are these nanochannels that I showed you earlier that fit these DNA molecules and force it to linearize. And because of that uniform linear stretch, we get all the information that we need just by taking a picture. We take these pictures, extract the digital molecules out of that, and then do a complete de novo assembly of the genome by pairwise aligning the patterns on every molecule that we imaged with every molecule that we imaged. We built a genome from scratch, and our software doesn't know that it's assembling a human genome or a tomato. We just built a genome that is there, and that's, of course, important. And then we can take these maps that were built de novo and align them to a reference or compare two samples with each other. You can do a tumor versus normal comparison or a primary tumor versus a metastasite, or we can do a child with a genetic disorder versus their parents. Now, Bionano has a few other applications. We really started off by building reference genomes. A lot of plant genomes and also animal genomes are built using Bionano. Our previous instrument, the IRIS system, was and is still used for that. Today, our speakers are mostly going to talk about modernizing cytogenetics and hopefully finding a replacement for some decade-old technologies, or many decades-old technologies, really. I'm going to mostly talk about novel SV discoveries, and I'll show you some examples of recent papers that show you how good we are at that. So to call structural variants, we simply compare these patterns of these labels on these maps with a reference genome of your choice. And when two label sites are moved closer together, we detect that as a deletion. We do that genome-wide starting at 500 base pairs. When the spacing between two labels increases, we call that as an insertion, and we call that at 500 base pairs genome-wide. We can look at repeat array expansions, and I'll show you an example that just looks like this, where we simply can count the number of repeats. We call tandem duplications automatically starting at 30 kb, direct or inverted. We detect translocations when a de novo map aligns for half with one chromosome and half with another chromosome, or a different part of the same chromosome. And we detect inversions when a pattern is simply flipped around, starting at 30 kb. So let me walk you through an example of what such an analysis looks like. We worked with the MD Anderson Cancer Center on a family where multiple children had an extremely rare, 1 in a million type of cancer. And they studied this family for over a decade with pretty much every method, every molecular technology that exists, and couldn't find an answer. So this family was mapped with Bionano as well, and I'm showing you just a piece of the genome here and some of the molecules, the very long molecules that we imaged. And you immediately can see that there's a pattern in these labels. So out of these labels, our software will build a de novo genome map, a consensus map, that represents the average spacing of these patterns. Now, when we align that to ESHG19, you see that the left half aligns beautifully. The right half has something going on. It's actually an expansion here of a 38 kb cassette that's present in 6 tandem copies. Now we built a second map because, of course, it's a diploid genome, and that map aligns perfectly fine with the reference. And in the controls that don't have this cancer, this duplication is not present. So what's going on here, this 38 kb repeat is perfectly easy to detect with our technology because we have single molecules that span the entire 230 kb of that repeat. We see that repeat on these molecules. We observe that structural variant. And that's very different than with sequencing, where you chop the genome up in little pieces and glue it together and hope that you did that well. We see structural variants on single molecules. They've since validated this with digital droplet PCR. And this gene is actually in the pathway that is known to be mutated in this cancer. So it was a big deal for MD Anderson. There's a genetic disease called FSHD, which has a very odd mechanism. It's caused by a contraction of a large repeat array subtelomeric on chromosome 4. And there's a pathogenic allele called 4QA and a non-pathogenic allele 4QB. And to genotype or diagnose this disease, you need to know exactly how many repeats there are, because if this collapses below 8 or so, the chromatin structure changes, a transcription factor becomes expressed, and this disease is caused. Now, to make this more complicated, there's an identical repeat on chromosome 10 that has nothing to do with the disease. With Bionano, it's very easy. You just look at it, you count the number of lines, and you're done. We distinguish between chromosome 4 and 10 because our molecules are so long that it anchors right into that chromosome, the rest of the chromosome. It's very easy to separate these. You can distinguish between A and B by the labels and just count each of these labels. There's a few published papers on this disease, including this one from Grandomics and a number of other Chinese institutes. And they're showing a mosaic case where the non-pathogenic allele is present with 19 repeats, and the pathogenic allele is present with 23, but also with 3 repeats. Because we have a single molecule technology, you can simply count the number of molecules that align to each of these variants to get an idea of the mosaicism. At this time, there's several companies and institutions who are validating our technology for actual clinical testing on FSHD. We're very excited about that, and we'll probably tell you more about that at ESHG if you come see us there in Houston. Now, typically, we don't have a label in each repeat unit, and in that case, we measure the flanking labels. And if the flanking labels expand, we have an idea of the size of the repeat expansion in between those labels. And that's something that was done here for myotonic dystrophy, where Eric Wang at the University of Florida wanted to correlate the exact size of the expansion with the severity of the phenotype. You see the normal allele here having a very distinct sharp peak, and the expanded diseased allele really has a very wide distribution. And so what we were able to show is that in a muscle biopsy, this repeat is unstable, keeps expanding, and so different cells in that muscle tissue have different sizes of that expansion. They purchased an instrument just to look at this type of disease. Now, those are really small regions, but we show you the large picture of the genome as well. Here's a part of chromosome 10. You see the centromere, or sorry, the entire chromosome 10. You see the centromere in the middle, short and long arm. And you can see right away that something's going on right there. So when we zoom in on that 7 megabase pair region, you see a large number of rearrangements, including these 2 inverted duplications. Putting something like this together could be a PhD project. It could be a career, but our software does this automatically in every genome that we assemble. This region is involved in intellectual disability. Now, there's really also direct clinical cases. Let's say you have a microarray call and you call a large duplication. It's important to know what happened to that duplication. Is it in tandem and disrupts a gene? Is it somewhere else in the genome and creates a gain of function? Bionano can show you that. Here's a case of a patient with a severe developmental disorder, and a custom microarray had called a 2.3 megabase duplication. When this child was mapped with Bionano, our copy number tool, which really works just like in NGS, an array in a sense, where we simply count the number of molecules aligning to the genome and normalize that. Our copy number tool detected 2 separate duplications here. But we add the order and orientation to this data. So now you can see where this duplication goes. It aligns here and also in this direction. And you can see that that second duplication is inverted, sits right next to the first, likely disrupts this large gene here. So we give you all that information that you just don't get with array. Now, I promised you some comparisons with sequencing. And in the last couple of weeks and months, 3 papers were published that did just that. The first one that I want to show you is a Nature Communications paper that looked at 154 human genomes that were part of the 1,000 Genomes Project. It's a large consortium where multiple groups perform high-depth Illumina sequencing on these genomes and call structural variants. Now, 154 from 26 different ethnic groups were also mapped with Bionano. And this was done years ago on our previous generation instrument with our previous generation labeling chemistry that had much lower resolution for SVs. And yet we call 8.5 times more insertions than Illumina could. 8.5 times. So if you're looking at a genome with just short-read sequencing, you just don't see those. We also saw about 1/3 more deletions, which you think are easy to pick up with sequencing, but not quite. And then just recently, a paper published by the Human Genome SV Consortium compared Illumina sequencing, PacBio long-read sequencing, and Bionano to look at the same genomes. And this graph here shows you the deletions on the left half and the insertions on the right half, ordered in size from 500 base pair here to 1 megabase in the middle. In this greenish color, you see structural variants that were picked up by Bionano and other technologies. But in blue, you have this entire section here of SVs that were only picked up with Bionano. If you use PacBio sequencing, assuming you could afford this, to look at a large-scale population of your patients, you miss all of these structural variants. Only Bionano can pick those up. Now, we get a lot of questions about nanopore sequencing as well. Long reads should be able to call these SVs quite well. So just this week, a paper came out in Genome Research from a group in Belgium where they looked at a reference genome at high coverage on their ProMethION and a read length of 20 kb. In comparison, ours is about 300 kb and 50. And because there's no good standard, they used 4 different SV calling algorithms on 4 different aligners. And they used this F variable, this F measure, which is the average of the sensitivity and the precision, so really the positive predictive value of these SV calls. And there's a lot of numbers here. I circled the best one, 70% of this combination of sensitivity and precision. The Bionano number of that variable is about 95%. So now, I'm just quoting from this paper. That means that there were thousands of variants that were missed and thousands of false positive called as SVs. Now, I'm not a clinician, but I do know that I wouldn't want to analyze a patient genome having to start with thousands of missing and thousands of false positive calls. That sounds like thousands too many to me. And their sensitivity for inversions is just 15%. They miss 85%, and of those, 70% are false. So it's just not usable. So calling structural variants genome-wide with clinical-grade sensitivity and precision is hard. We do it better, we do it faster, and we do it for less than any of the sequencing methods around. I showed you better. I didn't really mention fast, but with our improvements in the throughput of the instrument, in the sample prep, the labeling reactions, we can go from sample to results in just 3 days now. And we do it for less. The cost of a genome mapped at Bionano is just about $500 US. Now, all this applies to diploid genomes, standard genetic disease type of cases, constitutional disease. How do you think these technologies perform on complex, heterogeneous cancer samples? Well, I can show you how we perform. We just made significant improvements to the throughput of our instrument and a new chip that comes with it. So now, in a single run, instead of getting about 100x coverage on a human genome in 2 flow cells, we now can get 3 times 1,300 gigabase pairs. So 1.3 terabase pairs or 400x coverage on 3 human genomes in a single run. Now, if you collect this 100x coverage, which we recommend for constitutional cases, that means you can run 6 samples per day or 42 in a week. Our instrument can load 2 of these chips and move from one to the next. Or you can run these samples longer and collect 400x coverage on each of them. So that brings the cost per gigabase pair down to just about 39 cents, which is orders of magnitude less than any of the long-read sequencing technologies. And now we also released, just a few days ago, a new suite of software tools that can go in that high-depth coverage and pull out structural variants that we see in just a few molecules. And with that, we have the sensitivity now, 90% sensitivity, to pick up each of these major types of structural variants in just 5% allele fraction. That means if just 10% of your cells in your tumor have a heterozygous translocation, say, we pick that up. This is unbiased, there's no amplification. This is not a gene panel. This is any type of structural variant all over the genome at just 5% allele fraction. And to summarize what that means, no one else can do this. No one else comes even close to this. And we're not changing the cost, of course, so you get this type of sensitivity still for your $500 per sample. Our software shows you the entire genome analysis in a circus plot. Everything is interactive, so you can click on any of these translocations, zoom right into the de novo maps, and see the alignment with the different chromosomes, and then even show the molecules that aligned right to that variant. I want to finish by talking about 2 papers that were published recently, at least in bioarchives. The first one is by Jim Broach, who's head of personalized medicine at the Penn State University in Hershey. And he published a paper on about a dozen leukemia genomes that were analyzed with whole genome sequencing with Bionano. It's really worth reading because there's quite a few interesting variants. Here's one inversion, a 3 megabase inversion that was picked up in a non-Hodgkin's lymphoma, where the unaffected T cells were used as a germline control. And importantly, this inversion cuts right through P10, which is a known tumor suppressor gene for which there are about a dozen or so trials going on for compounds that rescue P10 mutants. So this is a structural variant that was missed by a whole genome sequence, that was missed by cytogenetics, and that could really affect the outcome and the treatment of this patient. And in this paper, they're showing a large number of structural variants that affect known leukemia driver genes. It's what you expect. But on top of that, there's also dozens of genes that are known cancer-associated genes, but not associated with leukemia, that have recurring structural variants. This is information that you don't get if you do a traditional cytogenetics on these genomes. This could have prognostic value, of therapeutic value. And on top of that, there's a large number of genes that, again, have recurring structural variants in these leukemia genomes that weren't even known to be cancer-associated. So there's an enormous potential there for biomarker discovery, for example. And then even going further, they also found that there's a significant enrichment in the number of cancer genes that are within 5 megabases or so of structural variants picked up with Bionano. These would be entirely missed with other technologies. These are structural variants that are maybe in regulatory elements, enhancers, that affect these cancer genes. There's a whole amount of data that still needs to be investigated. So Dr. Broach really believes that Bionano could replace cytogenetics, but we'll ask that opinion from our two next speakers that are going to talk about that as well. There's a webinar that he presented that's available on our website. I highly recommend that you have a look at that. Now, Dr. Broach manually combined Bionano data with Illumina data. There's a software solution for that as well from a company called Genox that has an annotation and classification platform for NGS data. The CEO of Genox is here. I recommend you talk to him later if you're interested in that. But what they do now is, instead of just randomly aligning the Illumina reads to call structural variants, which just doesn't really work, they use Bionano's SV calls to guide that alignment. So when we call that deletion in this region, they will use that information to guide the alignment of the short reads and automatically validate these SV calls with the Illumina data. And that way they can validate and refine the overwhelming majority of our SV calls with extremely high precision as well. All these variants are then annotated, classified using AI. Last paper I wanted to touch on is this one on the George syndrome, or 22q11.2 deletion syndrome. It's the most frequent genomic disorder. And in this paper, they show how Bionano can help elucidate these large segmental duplications, massive repetitive areas in this region that recombine to cause this disease. And using Bionano data on a large population, they were able to find different configurations and frequencies of these segmental duplications in different populations. I'm showing you this because there's a talk tomorrow afternoon at 1:30 from one of the authors of this study, and they're going to show this data, so check this out. And I want to just finish by saying that we have a blog called Bionano U. While we post presentations like this, discussions of papers, so please check this out. We have a number of posters. Several of those, the last 4, are tomorrow on molecular DNA isolation, on our cancer algorithms that I discussed, and also this one from the team in Edinburgh, where they also looked at a number of samples analyzed with cytogenetics and Bionano. And with that, I want to finish and move on to the real data right now.